Hi, I'm Terry Gaasterland. Thank you so much for the opportunity to be here. As you can see from my title slide, I'm going to be talking to you today about turning systems off, negative regulators in gene expression programs. I'll tell you a little bit about myself. I'm now the director of our Bioinformatics and Systems Biology program at UCSD. I'm a member of the Institute for Genomic Medicine, and my home department is the Scripps Institution of Oceanography. So you're seeing here a view of what I'm so very, very lucky to look at every day, and I thought I'd share it with you. And you're seeing behind me in my scenery, Scripps Pier from the Scripps Institution of Oceanography. I'm going to split my talk today into two parts. We're going to talk about turning systems off and looking at this, how important the breaks are in biology. And the first is going to be a stem cell talk. We're going to talk about developmental signaling pathways. And the second part, which seems to have been labeled 1 here for whatever reason, is going to be negative regulation in prevention of neuronal cell loss. So that it will be a disease-oriented talk. And the first one will be a normal systems-oriented talk. So to get us all onto the same page, in many ways, we can think about development as organization, as specifying what's on top, what's on the bottom, what's left and right, front and back. And I like to think of this as organizing the first 24 hours of life, whether we're a mouse, a human, a sea urchin larvae, or a sea urchin adult. Producing that organism is the process of organizing that first 24 hours. And I say the first 24 hours because I was inspired early on, back in the 2000s by Eric Davidson and his work in sea urchin. And what we're seeing here is a circuitry that he laid out for the early development, the first 24 hours of sea urchin. And he always liked to say the first 24 hours of life for a sea urchin is equal to the first 6 weeks of life for a human. So going on, I'm specifically going to talk to you today, about Wnt signaling. And Wnt signaling is really a very special thing. It controls the head versus the tail during development. And as it turns out, looking at this tadpole here, if we were to measure Wnt expression levels from the very tail to the very head, we would see that the expression of this gene and the protein made from this gene is high in the tail and lower in the heads. And in fact, if we take a planaria, and here's the head with the 2 little eye spots and the tail, and we knock out Wnt, that planaria has 2 heads. And if we upregulate Wnt, the planaria will develop with 2 tails. So we can manipulate this. And in fact, there are situations where you can have an adult organism that can be induced to have extra heads. This is a little schematic down here where we can see the Wnt protein molecule, and there are 19 of them in the human genome, interacting with the LRP6 transmembrane molecule and the frizzled 7, or the frizzled transmembrane receptor. And there is a whole family of the frizzleds. So there's a many-to-many interaction of the Wnts to the frizzleds. And what we're seeing here is the core mechanism by which Wnt signaling starts. And summarizing over here, Wnt signaling specifies the tail. Wnt is high where the tail is. And defective Wnt signaling leads to severe developmental problems. Focal dermal hypoplasia is just one, but all sorts of cancers are related to Wnt. So let's step back and ask the question, for a developmental signaling pathway, what are the components? How does that pathway run? Well, it needs to be turned on. It needs to progress, and it needs to end so that we can set up the situation for the next step, the next pathway to be invoked. And many pathways follow this paradigm: Wnt, FGF, hedgehog, TGF-beta, pathways involved in integrin signaling, Notch. So these are all specific instances of what we call developmental signaling pathways. So we're going to talk here about Wnt. So let's think about, how does Wnt work? I showed you that sketch in our primer slide a couple ago. Well, beta-catenin is a critical factor for Wnt, and so is this destruction complex. This sits inside of the cell in the cytosol. We're looking at the outside of the cell boundary here, and we're looking at the nucleus boundary here. We have the DNA down here with repressors sitting on the DNA and transcription factors that are ready to go and activate genes. Well, can they get activated? If another cell expresses Wnt, so this is another cell nearby, and that Wnt binds to the frizzled, the destruction complex gets recruited, it's no longer available to gobble up the beta-catenin, and the beta-catenin is able to go across the cell nucleus membrane, bind, co-together with these other transcription factors represented by the green here, and make those target genes go. So that's in a nutshell how Wnt specifically, and this cloud up here, this destruction complex, is the critical factor that gets recruited. So let's think about termination of Wnt signaling. There are 3 different ways. Those target genes that I just showed you invoke the expression of Wnt antagonists. Some of them go and target Wnt itself and make it not be there. Others target in this Wnt to beta-catenin activation area, and others inevitably, well, others target beta-catenin itself. And then finally, we want to know, are there genes that are activated by Wnt that actually turn off the other genes that are activated by Wnt? That's been an open question, and I'm going to tell you about our detective story where we explored that through high throughput sequencing experiments. So criteria for a terminator of Wnt signaling. Well, we're going to look for things that are highly induced in response to Wnt. So it's got to go up, and it's got to be off and then go up. We're going to look for transcription factors. So these would be transcription factors that are activated by Wnt and go and repress the genes that are activated by Wnt. And we want them to act globally, not just one gene or another gene. This should be turning off the entire program. So we can hypothesize there is a gene like this, and we can find it through RNA-seq. So let's look. Well, first of all, let's set up our system. So here we have at 0 hours human pluripotent stem cells that are treated with Wnt, in this case Wnt3A, for 12 hours, 24 hours, and 48 hours. And what you're seeing here is the cells are changing, and over that 48-hour period, they're organizing. So this is our experimental time series and our platform. Well, we did RNA-seq at every one of those time points and analyzed the data. So here we have 0, 12, 24, 48, and we organize the data to look for genes that are on and then turn off. So the blue here corresponds to 100% of expression at their maximum time point. And then here we have genes that are off for the most part or low, and they turn on subsequently. We go a little further and we have genes that are even more off, and they go even more on at the 24, 48-hour period. And then at the bottom down here, we have genes that are very much off and they're very much on at 48. So we call these transiently upregulated, and late upregulated. And as it turned out, when we did this experiment, it was quite heartening to see, excuse me, that these transiently upregulated genes, were genes involved in mesendoderm. For example, we see T, brachyury, nodal, MYCL1. The slightly later upregulated genes that stay up, slightly, I say, in the 24-hour period, were genes that are known to be organizing the primitive endoderm. And then these late upregulated genes included SOX17, DKK1, and these are genes involved in definitive endoderm and tail budding. So we did this RNA-seq experiment. We did a time series. We did not know what we would find. And sure enough, out of the RNA-seq data, when we organized it by this time series, we had a result that made sense. Not just that, we then took the question and asked, what transcription factors do we see here? And sure enough, SP5 popped out. SP5 is one of a family of SP transcription factors, and it was even more upregulated than axin-2, which is the transcription factor that has been most popularly studied in Wnt signaling. And if you talk to the Wnt community, axin-2 will be the first transcription factor on their lips. So SP5 was the surprise. So our next step was to really look at it. Well, here's SP5 in relation to axin-2. So you're seeing here on the y-axis that SP5 is going way up more than the axin-2 did. This is through qPCR. We were able to see that axin-2 peaked at 6 hours. So this was even before our 12-hour time point. So our view of axin-2 was without the 6-hour peak. And SP5, in contrast, just started going up and up and up, and by 24 hours was way up. And over here, we took a look at the SP5 protein expression, and you'll see in this gel that it goes again up and up and up. So whether at the RNA level or at the protein level, we're seeing SP5 going up. Well, I told you that SP5 is one of a family. SP1 is a transcription factor like SP5, and I'm going to show you the domain architecture in a moment here. But SP1 is ubiquitously expressed, and that's going to be very important. And our protein time series here shows that SP1 is present and expressed in these cells at all time points. So let's take a look at SP5. Here's SP1. This blue domain in this cartoon of the protein shows that it shares the BTD domain and the zinc finger domains with its cousin SP5, but in contrast, the SP5 gene has a domain, has coding regions for a domain of the protein that's involved in repression. So in other words, SP1 and SP5 are the same for the part that bind to the DNA, and they are different for the part, for a different part here. So that we'll come back to that. So remember that architecture. So to show that it's a transcription factor and that SP5 is indeed in the nucleus, Ian Huggins, who was the experimentalist on this work as a graduate student, along with Carl Willard, did a series of experiments where he actually imaged where does this protein get expressed using fluorescently labeled antibodies. So we wanted to know, what genes does SP5 target? Well, ChIP-seq can give us that answer. And what we're seeing here is, let's see, SP5 bound to DNA. We've shattered the DNA, and we have antibodies to SP5. We have one binding here, and having an antibody to the SP5 enables us to be able to pull this out, immunoprecipitation, and then sequence the DNA that's bound to the SP5. So that's ChIP-seq in a nutshell. Well, we did this, and we took all of our reads from the ChIP-seq, so these are high throughput sequencing reads, and we mapped them back to the human genome. And then we blended the reads and called peaks using a peak calling tool. And what we see here is SP1 peaks in untreated cells, SP1 peaks in Wnt treated cells, SP5 peak in untreated cells, and SP5 peak in Wnt treated cells. And the pattern we have in this particular gene, which is PLK4, I picked this gene because it just has an incredibly strong and clean pattern, is SP5 through these reads that led to these peaks is binding strongly under untreated cells. And that SP1 binding goes down, I'm sorry, SP1. SP5, in contrast, has hardly any reads, and then it goes up in the Wnt-treated cells. So this is telling us that in the exact same place, SP5 is coming and binding, and we can hypothesize again. Notice the y-axis. SP1 is at 120 here, SP5 at 50. That's peak height. a normalized peak height over numbers of reads mapped to the whole genome, and then for size of the peak. So we have SP5 potentially displacing the SP1. Well, we gathered all these peaks from across the entire genome for every gene where we had SP5 peaks and SP1 peaks, and we used a pattern generator to look for all of the patterns that emerged from under the sequences across all of the peaks across all the genomes. And sure enough, the SP5 peaks had a pattern that was very the same. This is called a GC box, very much the same as the SP1, with one exception. The SP1 sites were enriched in CNA, and that was the SP1 peaks across the whole entire genome. Remember, I told you SP1 is ubiquitously expressed. Well, it's also ubiquitously bound to GC boxes in front of just about every gene in the genome, 20,530 of them, where SP5 was much more selective. SP5 was binding in just 475 places. So acting globally in the sense that we had almost 500 genes, but quite selective in the sense that it was only 500 out of these 20,000. And sure enough, the SP5 pattern was the subset of SP1 peaks that were enriched in an A in this position. So that told us that there was something special in the genome, and there is a motif, incredibly subtle, that influences the recruitment of SP5 to one gene or another. We now took that canonical pattern, high, not so high, pretty much off, turns on, and you'll see here we have a region in axon 2 where SP5 is binding, but the peak patterns remain the same. Amodal 1 has in fact 2 binding sites for SP5, and so on. So I'm showing brachyury is another one. Remember, axon 2 and brachyury T are genes that turn on and then go back down. So SP5, as it's coming up, and I'll go to the next slide. Whoops, I didn't have it here. And I didn't have it here either. Oh, I'm so sorry. Well, SP5 is also self-regulating. So SP5 turns itself off in the exact same way here. So an open question in all this work is, how do these different peak heights and binding affinities govern how some of the genes go up early and then come down, and other of the genes take a little while to go up and then stay up? So different affinities for the SP1 versus SP5, open question. So now we know SP5 targets, in fact, we were able to mine a little bit more. We had 450 genes in that pattern before, and we confirmed that SP5 targeted 662 genes. We want to know what happens if SP5 isn't there at all. So Ian created these CRISPR-Cas9 constructs and transfected them into cells, and was able to build 2 cell lines successfully that had SP5 edited out, deactivated by this CRISPR-Cas9. And we use those cells to do more RNA-seq. Oh, and here, this should have been next. So here's the SP5. So you can see that SP1 is actually binding across the entire SP5 gene. And under Wnt activation, it's going down quite a bit. And SP5 untreated is hardly binding at all across the gene. And here's that peak of SP5 getting ready to turn itself off. So SP5 is exactly what we knocked down. So here's the time series. SP5 wild type, so exactly the cells that I just showed you at 0, 12, 24, 48. So these were a new control. This is our baseline. And then our SP5 knockout clone 1 and a different SP5 knockout clone 2. So we effectively did the knockout in duplicate in 2 completely separate ways. And what we're doing here is coloring 0 to 100 again. So 100 is blue for 100% of the maximum in the regular normal time series. And then we go on up to 200 or above for any gene that goes higher than the normal. And what we saw here was that, in fact, many of these genes that fail to reach a peak and go back down, or reach a height and stay there at some stochastic level, just kept going up and up and up. So this is representing those 662 genes that we found to be bound by SP5. So especially the late regulated genes, defining definitive endoderm and tailbud, just went up and up and up even more with the SP5 knockout. So we also wanted to ask the question, what do these cells look like when they're untreated and they're simply left to grow? Are they normal? Or if they're not Wnt treated, do they look okay? So here we have wild type, the clone 1 and clone 2, at day 1, day 4, day 7, and day 10. And sure enough, these clone knockouts are exhibiting what we might expect in retrospect. They're getting bigger, they look abnormal, these are kind of monster cells, and this is solely due to that SP5 being knocked out. And over here in these genes, we can take a look and see that FOXA1, MIXL1, SOX1 are abnormally high in these blue and dark blue qPCRs in these SP5 knockouts. So this shows that these genes are, these cells are just absolutely strange genes when SP5 is gone. So we have found a critical regulator. Wnt induces many genes. Those many genes include SP5. And of those genes, SP5, by our ChIP-seq experiment, we were able to confirm targets 662. And we asked what happens. Well, SP5 reigns in, it caps the maximum expression level of those genes that it controls in their response to Wnt. And I'll just go back and point out that it's not all of them. So it's about half of them that really have this high level. However, if you look, there are certain genes that even at their baseline are higher than the maximum over here. So these are genes that are already upregulated. So SP5 at some very, very low level at the very beginning is involved in keeping them at that lower level. So SP5 is somehow balancing these 662 genes and their expression levels even before the Wnt starts, and especially after they receive the Wnt3A signal, Wnt3A in this case. So here's the model that we can put together from this. We have initiation, Wnt comes along, it signals to beta-catenin. Beta-catenin doesn't get destructed and crosses to invoke target genes, of which SP5 is one, Wnt antagonists are other. So in the progression stage, these Wnt antagonists start shutting down the early signaling so that new signals can't come in. SP5 continues to be expressed. And then in this termination stage, that's when SP5 is most important in turning back off a subset of those target genes that have been turned on. So I want to give you a vignette now on top of this story. Shortly after we completed this work, a paper appeared of some work in Hydra. This is Vag Bakari et al., published in Nature Communications in 2019. This was not our work, but it was a really great cheer for our work, because what they looked at was SP5 in Hydra. And they did the knockouts and they did the upregulation, and they realized that this SP5 feedback loop that I just told you about that we'd worked out in human restricts the head versus tail organizing activity in Hydra. So I'll give you the heart of what they did. So here's the wild type Hydra. Wnt gets expressed, and we have the beta-catenin Wnt3 cycle. Beta-catenin induces SP5, and SP5 turns around and stops the Wnt-induced genes, just as I was telling you here. So we filled in this part. When SP5 was restricted by RNAi experiments, the intact Hydra all by itself just had multiple heads, just like I showed you on that very first slide where I was introducing the Wnt. And in the regeneration experiments that they did in this paper, multiple heads came back. And they also had a reaggregation with multiple heads aspect to this. So that was with the SP5 knocked out, where this feedback was knocked out. So this was a Hydra experiment that recapitulated what we had noticed in the stem cells and really nailed it, that with SP5, you wind up getting multiple heads. So SP5 had this unexpected outcome. So what did I just tell you about? Wnt signaling controls head versus tail organization. We did RNA-seq, ChIP-seq, and CRISPR-Cas9 experiments, followed by more RNA-seq. And the data from those experiments revealed SP5. It told us that this is a new transcription factor in Wnt signaling. It's a negative controller. It terminates the Wnt signaling. We saw that stem cells with no SP5 grow abnormally, and Hydra with no SP5 grow multiple heads. So even the Hydra was an unexpected phenotype, but demonstrated that the SP5 is really critically important all the way from human to Hydra. So that was the story about organizing the first 24 hours of life and looking at Wnt development and a critical new transcription factor in that developmental pathway. It was a pleasure and a surprise to find that there really was a transcription factor there, and it wasn't this big complicated multi-way organization, but we were actually able to pull out one repressive transcription factor that made all the difference. So now I want to shift gears and go to this second story. I want to tell you a little bit about work my lab has done in looking at negative regulation in the prevention of neuronal cell loss. And in particular, we're going to look at retinal ganglion cells. Retinal cells are the top layer. This is a cartoon of a human eye with a cross-section. This yellow area here represents the retina. And the very outermost, so this is the back of the eye with a choroid, and this is the front of the eye. These are this top layer on the very edge here. These ganglion cells are the cells that receive the signals from the photoreceptors, and their axons bundle together and go backward. Well, they actually go sideways. So they're coming sideways all the way along the retina, and then they're taking a 90-degree turn here at the optic nerve and bundling together to create the highway that's going to take signal for vision, all of those photoreceptor signals, to the brain, to the visual cortex, so that we actually see images. What we're seeing over here is what's called a fundus or a fundus photo. And we're looking straight, I can't point to it, we're looking straight through the eye here at this area, which is called the optic nerve head. What we're seeing in this photo is this little notched area here and this lightened area here, where this particular human being, this patient, has lost those axons from those retinal ganglion cells. And they've lost them through high pressure in their eye, inducing retinal ganglion cell death through apoptosis. So an open question becomes, can we find a way to protect these retinal ganglion cells? Can we prevent optic nerve loss in high pressure glaucoma? Well, right now, the only treatment for doing this is to lower the pressure in the eye with eye drops that slow the development of, or the generation of aqueous liquid inside the eye. So to try to answer this question, I participated in a genome-wide association study together with hundreds of others. And the group was called NEIGHBOR, the National Eye Institute, so that's in the United States, glaucoma study. And the NEIGHBOR study, under the leadership of Janie Wiggs at the Massachusetts Eye and Ear Institute at Harvard, she was able to assemble slightly over 2,500 cases, 2,500 controls, do classic genotyping by Illumina 660 chip, and the group did the GWAS to identify overrepresented SNPs or underrepresented minor alleles of SNPs and find these pileups. So I'm not going to go into genome-wide association studies other than to say we can take every SNP in the genome and organize it from chromosome 1 to chromosome X and graph it. This is effectively a scatter plot of where in the genome does it occur versus what is the p-value, what is the negative exponent of the p-value, which gives us the likelihood that this particular SNP, one allele, is overrepresented in cases versus controls. And what we found in this genome-wide association study was, sure enough, there was this region with all these SNPs that piled up as overrepresented for one allele or the other. There was another site over in the neighboring chromosome for the 6166 region. So these were the two areas that were pointed to, and there's been some work looking at these genes in the ensuing years since this was first published in 2012, but very little has broken through mechanistically. So what I want to tell you about here is a bioinformatics experiment followed up by a very simple in-lab experiment that we've done in my lab. This is the work I'm about to tell you is not published yet, but I felt it was important enough to give you this theme of the negative control. So let's take a look at what we did. So here's a UCSC genome browser view of this particular region. Here's the CDKN2B gene, and here's this other gene, CDKN2B-AS1, an antisense gene. And this happens a lot in the genome, where you get this long, weird, non-coding RNA, and its transcription is actually starting here. It's being transcribed in that direction. And in fact, the CDKN2B gene is in the first intron of this antisense gene. So we can think of the antisense gene flanking and bounding the CDKN2B gene inside of this. As it happens, there's a CDKN2A gene, so a cousin of this one, a paralog, that shares a promoter region right in here with the antisense gene here, and CDKN2A is transcribed in that direction. So both of these are inhibitors, that's the N, of CDK, cyclin-dependent kinases. They're the second inhibitor, and in this case, in the second family of inhibitors of CDK, it's the B and the A. So CDKN2B, a little bit of gene nomenclature. And I've sketched up here, CDKN2B antisense, CDKN2B, CDKN2A to show you the directions. So one of the SNPs in that pileup, and I'll just go back, so here's that pileup, this yellow here. Well, each one of these is a SNP with an RS number in the human genome and the dbSNP database. Well, the SNP that conferred the most risk, actually the only SNP whose minor allele was overrepresented and was overrepresented thus in the risk direction, all the others, the minor allele was underrepresented, so the major allele was overrepresented, and I'll leave that as a puzzle to think about for some of the more computational people in the lab and the people in the audience and the people who think about recombination. But here's the SNP, RS3217792. It's the flagship SNP in the main table in this particular publication of our GWAS. So we said, you know, that SNP is in the 3' UTR. Here's that first exon. Remember, transcription's going this way. Here's the second exon. And the SNP was pretty much smack in the middle of the 3' UTR. So we took just that sequence. We used the UCSC genome browser. You know, we zoomed in on it. We took just the sequence. And here's the sequence. The sequence is going from here to here in reverse. So we took that 3' sequence and ran it through a tool that we had built in the lab, Zoom Mirror. And what it does, it takes a query sequence and matches it with imperfect mismatches against the microRNA sequences, the human microRNA sequences available in the microRNA database at the Broad Institute. Where is the microRNA database? I guess it's at EMBL at this point, EBI. So here's a sketch of a microRNA. Transcribed before it's processed, it forms a hairpin. And we have this stem where the microRNA, the premier, is cut, And you'll see that there's slight mismatches between the microRNA, the front part, and then coming around, and this back part. So we might call this a hairpin or a lollipop. Well, what's most important is both sides of this can get processed into a mature 21 base piece that can turn around and do this. It can bind to an mRNA. So here we have the 5', the beginning of the microRNA, to the 3'. So we're creating a double strand here. And over here, and one more thing, I'll just talk about nomenclature. So miR-138 is this microRNA. The 2 talks about this, the 2 is that it's the second copy of this in the genome. And the 3P, which can also be represented as a star, is that it's this purple arm. So the 5' arm and the 3' arm. So this 3' arm is what we found to bind potentially through our Zoom-Mir computational predictor that Shujie Wang built originally for Arabidopsis thaliana, and that we've now adapted to be able to do this kind of screening in eukaryotes that are not plants. So to adapt it, what we did is say, well, let's not screen, unlike in plant where a microRNA has to bind the entire beginning to end. Let's just restrict it that we'll just look at the first 8 bases of seed. And what we found was in this first 8 bases of seed, RS321-17792 was a SNP in the second to last seed site. So the C here was turned into a different nucleotide in the minor allele. So this raised the question, all right, can we do an experiment where we can check both alleles and also check a fully scrambled seed, so this whole 8 base is completely scrambled, and also check a perfect binding of the microRNA. So here's what we did. So we took we synthesized the microRNA itself, and then we inserted we synthesized the mRNA with certain constructs in it, and then we synthesized it with the alternative allele, and we synthesized it with the perfect seed over here. With the totally disrupted seed. That's what the X's are. So what did we find? That in the natural mRNA, with different doses of the microRNA, the mRNA went down. So here's our negative control in the black, and in the blue, the mRNA expression levels go down under increasing doses of the microRNA. And in fact, it didn't take all that much to get most of the effect. Meanwhile, over here, when we introduced just that one SNP, the one base change of this RS number, we lose that signal entirely. Likewise, when we disrupt the seed entirely in this mRNA target, we get a similar pattern to just the SNP alone. So what this told us experimentally, and this was done with Amy Dubinsky, who has now gone on, and she's now at Illumina in their genomics laboratory. What this told us is that just that one base change was as strong an effect as changing all 8 bases of that microRNA, effectively obliterating the ability for the microRNA to bind to the mRNA. So then we turned around and said, well, what else? So using our Zoom-Mir tool in reverse, let's take the microRNA as a query and ask what mRNAs out there are also targeted by this particular microRNA that we have demonstrated to have this strong ability. So it still remains to be checked in cells themselves, but we checked it with this luciferase assay. What else does miR-138-2 3' or star bind? And what we found was here it predicted CDKN2B. That's a good thing. It recovered what we expected. We also got a really strong signal, and these are the scores over here, for ataxin 1 and ataxin 2L, which is interesting because subsequent GWAS papers that have done what are called meta-level GWAS, they've integrated multiple genome-wide association studies to up the number of patients from 2,500 to many thousands more, have revealed ataxins as one of the genes involved, associated with glaucoma. We also found the 6-1 gene, which was that other peak over here in the green area in our original GWAS. So 6-1 has a control site. So what this demonstrates to you, or what this illustrates, is this ability to take a bioinformatics approach, add some experimentation to confirm what we learned through bioinformatics, and then turn around and expand our bioinformatics approach to learn more. So we took our SNP, we looked for microRNAs, we found one, we confirmed it, at least with the luciferase assay, and then we asked, what else does that microRNA potentially do? So the pattern here that I wanted to bring out is that this microRNA has potentially at least 3, ataxin, CDK, CDKN2B, that is, and the 6-1 targets, all of which are genes that have been associated with primary open-angle glaucoma, a blinding disease that leads progressively and inevitably to blindness in people. So now we can put this together into a model. So we've got our SNP and with its 2 alleles, and we have lower risk when we have the major or the more frequent allele in the population, and we have higher risk for glaucoma, we know that from the GWAS, with the minor allele. And now we can fill this out. Well, in that lower risk, our microRNA binds the mRNA degrades, the CDKN2B, and also there was one more gene that I didn't tell you about in this particular version of the story, but CADM2 levels go down, and then cell cycle is uninhibited. CDKN2B, the inhibitor of cell cycle, doesn't inhibit anymore. And what we know is that its target, CDKN2B, is now allowed to go through G1 progression. And I put a Wikipedia cell cycle cartoon here because this part of it, the fact that CDKN2B controls cell cycle, is well known in the literature. Well, over here with the unbound mRNA, so the microRNA isn't turning off the CDKN2B. We make the protein, we wind up with increasing inhibitor levels, and so say cell cycle gets inhibited, and we wind up not having the G1 progression. So the question becomes, what is the key type of cell in the optic nerve or in the retina where this is happening? So the next slide, I'm going to do a little dive into the literature and expand our model simply based on a literature study. So putting the parts together in the eye. So what we know is that angiotensin and TGF-beta under stress, including pressure in the eye, induce the SMAD transcription factors. That is known. What we also know is that this SMAD binding response element winds up not being occupied by SMAD. In the SP1, here's SP1 again, MYC-MYZ complex upstream of P15. And I want to show you here that P15 is a nickname for CDKN2B. So when SMAD gets expressed under pressure, it binds to this response element, and it binds to the SP1 and MYZ, displaces MYC, and now the CDKN2B gets expressed. So the stress expresses the SMAD, which invokes the inhibitor of cell cycling. And here's the cartoon from this particular paper where we could put it at a higher level that MYC represses P15 in the presence of MYZ, and P15 represses CDK4, CDK6, which are the cycle-independent kinases that push through the G1 part of the cycle. Well, what we also know from the literature is that angiotensin and TGF-beta1 upregulate SMAD2 and 3 at the lamina cribrosa, and not just that, we also know from the literature that they upregulate CDKN2B. So CDKN2B, as we see over here, is inhibiting the cell cycle. And so the open question is, and we've shown here in the experiment that I just showed you, that MIR138 inhibits the CDKN2B. At least I've shown it mechanistically through the luciferase experiment. And a big question is, what regulates this microRNA and helps potentially mediate within this pressure response? So the hypothesis is now that people who are more susceptible to glaucoma have less binding of this microRNA to the inhibitor cell cycle. And that is the critical explanation for why they wind up with more optic neuropathy. So I'm going to leave this story there. And the hanging question is, what cells is this happening in? So that'll be our the next part of this story. So the highlights that I've just told you about are on preventing optic nerve loss. We had a risk SNP for primary open-angle glaucoma, POAG, that was found through genome-wide association using a little over 2,500 cases and a little over 2,500 controls. That was reported in 2012. And here's our one SNP whose minor allele, the allele that is less frequent in the general population, is overrepresented in our patients with the disease. This SNP is in the 3' UTR of this cell cycle inhibitor protein, CDKN2B. The SNP disrupts microRNA binding. I showed you that with the luciferase assays of this microRNA that we found through our computational tool, Zoom-Mir. And here I put the star instead of the 3P. So this sets the baseline for the hypothesis that this risk allele is somehow leading to reduced cell renewal. So the open question is, what kind of cells? It's certainly not neurons themselves, because neurons are terminally differentiated. So what is it that's going on right here in this area in people who express this microRNA and have it bind to the CDKN2B in a normal way versus disrupted by this SNP? That's the next chapter to come. So I've told you two stories about turning systems off. Negative regulation in developmental signaling pathways. We looked at Wnt and our discovery of the SP5 transcription factor and its critical termination of the program that gets turned on by Wnt. And then I've told you about negative regulation in prevention of neuronal cell loss. And there it's the CDKN2B and the MIR138 story. So I'll give you some acknowledgments. Ian Huggins was the graduate student. He's now a postdoc who did much of the experimental work in the Wnt stem cell system. That work happened in Carl Willard's lab at UCSD. He's part of the stem cell program. In my group, Lee Edsel, Tessa Pierce, Simar Petros, and Bailey Shabbat are bioinformaticians who have worked on this data and getting it going. And we do all of our sequencing over at the Scripps Research Institute's Genome Center, and that's run by Steve Head. And I have to give a hats off to Lana and to Phil for the heavy lifting. And if you'd like to get follow-up reading on what I told you about, especially about the stem cells, just Google, well, do a search at NCBI on Gaasterland T and Willard K, and you'll see the various papers that we've written, our two labs together, the Willard lab and the Gaasterland lab, on this particular area. And this is sunset at Scripps. I started with a sunrise, ending with a sunset. And I want to thank you for listening. I'm hoping that we'll work out between now when I'm recording this talk for you and the day that you're looking at it, that I'll be available to do some Q&A live, because this work I find incredibly exciting. It really grounds both, you know, how do we organize life at the very beginning, and how do we keep using these regulators, these critical negative regulators over time, over the lifetime of our body, to prevent really bad things like going blind from happening. Thank you very much.